Okay, so I'm sorry about Friday's lecture. Like the file got corrupt in some way and nothing will open it. So I think it's pretty good. We made it all semester without that happening. Um, so we started with just learning about different methods for studying apoptosis, about caspases, introduced intrinsic and extrinsic pathways of apoptosis, and now we started talking about BCL2 families of proteins. And BCL2, a good way to remember it is because it comes from B cell lymphoma. Because when there's a mutation, cells tend to not induce apoptosis. If you don't induce apoptosis, you tend to become a cancer cell. Okay. Do you like this voice? It's nice. Okay, so let's just talk a little bit more about the BCL2 family proteins. Should be a fairly short lecture because I started trying to think, what should I focus on with cancer? And I was like, there's way too much and it's the last bit of class. So we're just going to finish up by apoptosis. You guys are going to understand that a lot of mechanisms of... People are walking by laughing at me. Many mechanisms of apoptosis are inhibited in cancer. So if you can't, if you have dysregulated cell cycle and can't induce apoptosis, you have cancer. And even worse, some cancers have mutations in p53, which is a big inducer of apoptosis. And not only do you end up with cancer, but you can't respond to chemotherapy because a lot of chemotherapy depends on inducing apoptosis. So, not good, not good. Okay, so with the intrinsic mechanism of apoptosis, these are regulated by the BCL2 family of proteins. So if you remember the extrinsic mechanism of apoptosis, you don't really have any BCL2 proteins. It's just you've got a death receptor and it gets ligated and you activate the initiator caspases and then activate the executioner caspases and then you've got apoptosis on its way. It's only the intrinsic apoptosis. Only the intrinsic apoptosis that uses the BCL2 family of proteins. Okay, so we're going to start with a movie which puts it all together. And I believe there's no sound except for just like sound effects. And then lecture 1, 2, and 3. This is a series of 3 lectures done by someone who's an expert in apoptosis. So if you are like, what is she talking about? Take a look at these lectures and see if you can pull something out of there. It's a guy who pretty much discovered the role of cytochrome C in the intrinsic pathway. Okay. Let's see. Why? Give me 1 second, guys. Now we're going to cry. It can be saved. Don't worry. It can be saved. Okay. Hold on here.
The world's really against you.
Huh?
The world's really
That's against me. You know what? That's fine.
Thank you.
There we go. Oh, God. No one uses this. It's really dusty. And it's good everyone's late today. Okay, while we wait for that to start up, we'll come back to the movie in a minute. Okay, so let's just go over the role of these BCL2 proteins. Remember, BCL2 is a family of proteins, and there are 3 categories. There's these BH1, 2, 3 proteins. Those are the pro-apoptotic proteins that actually form the pore. in the mitochondrial membrane. And then there are the anti-apoptotic BCL2 proteins that inhibit the BH123 proteins. And then there are the BH3 only proteins that induce apoptosis. So let's go back for a minute. So the BH123 proteins, the most popular that people talk about are BAX and BAC. They oligomerize, so that means they bind to each other and they form a pore in the outer membrane of the mitochondria. And so that's what that pore is what allows cytochrome C to come out of the mitochondria. It's also referred to as mitochondrial outer membrane permeabilization, MOMP. And this results in release of cytochrome C from the intermembrane space. So this leads to the question from this weekend's quiz. So let me take a quick poll. How many people knew there was a quiz this weekend? Because I mentioned it multiple times in recitation. Okay, because there are a handful of people who did not take it. Okay. Okay, a defect in BH123 proteins, such as BAX, will likely result in all of the following phenotypes except. So this should be pretty easy because one has nothing to do with the intrinsic pathway. Which one is? Which one? D. Yep, the disc that disc is involved with the extrinsic pathway of apoptosis. So cytochrome C, right? BH123 protein. That's the proteins that oligomerize and form the pore and allow cytochrome C to be released. If that doesn't work and you can't form a pore, cytochrome C won't be released, right? Upon treatment with X-rays. Remember, DNA damage from irradiation induces the intrinsic pathway. Apoptosome will not assemble because cytochrome C release that results in formation of the apoptosome and procaspase activation. So A, B, and C will not happen if BH123 proteins don't work in some way and can't form the pore to allow cytochrome C to be released. Okay. Give me a minute. Thanks for your patience today, guys. Okay, so if there's no apoptotic stimuli such as DNA damage or drugs or whatever, then there are these anti-apoptotic BCL2 proteins, and they will inhibit the BH123 proteins. So they actually prevent the oligomerization of those proteins so you don't have cytochrome C release. When you induce apoptosis, BH3 only proteins, so this is BAD and BIM. You can think of BAD as, well, it's bad, right? And because it's going to induce apoptosis. Well, bad, no, inducing apoptosis is good sometimes. But anyways, they bind to the anti-apoptotic proteins and allow this oligomerization. So the way to really think about it is just, do you have the pore forming or not? And it all has to do with whether or not these proteins can oligomerize and form the pore, and that's all regulated by whether or not BCL2 can inhibit or not. So this, if you have apoptotic stimulus like DNA damage, then BH3 only protein will be activated, and that will bind and inhibit this BCL2 protein. So that allows this to be oligomerized, and cytochrome C will be released. Okay, let's pull this up. Okay, so then there are the IAPs, inhibitors of apoptosis. They do exactly that. They bind and inhibit activated caspases. So let's say you have caspases that just become activated spontaneously. This can happen. There's a way to prevent that from triggering apoptosis when it's not supposed to happen. And these are IAPs. And all they do is they just bind to they bind to, so here's your procaspase. Something will happen that causes activation, so your prodomains will be cleaved, and then they become active. IAPs will bind them and prevent them from working and activating other caspases and promoting apoptosis. Okay, which of the following apoptosis regulators are analogous in function? So what I meant by this question is pro-apoptotic versus anti-apoptotic, right? Which ones are pro-apoptotic or which ones are anti-apoptotic? So IAPs, are they pro or anti? Anti, right? Because they prevent caspases from functioning. BCL2, was this pro or anti? Anti, because they prevent the oligomerization of the BH. Here, go back to this one. Right there. The BH1, 2, 3 proteins can't oligomerize. Anti-IAPs, which we're going to talk about in a minute, those prevent IAPs from functioning, so those are pro. And fast ligands, you guys remember fast ligands? That's pro, right? Because fast binds to fast ligand and signals and starts the extrinsic pathway of apoptosis. So the answers were A and B. Okay, so anti-IAPs, those actually are inside the mitochondria in the intermembrane space. So when the pores forms and cytochrome C is released, other things are released too, one of which is anti-IAPs. And they do exactly that. They are anti-IAPs. So they block the function of IAPs, and that allows apoptosis to happen. That's kind of logical, right? Because if you release your cytochrome C from your mitochondria, you're trying to activate apoptosis. You're trying to activate caspases. So you want to prevent your IAPs from working. Right? So you have your stimulus of apoptosis, your BH1, 2, 3 proteins. They oligomerize, form the pore, release all this stuff, including cytochrome C, which activates the apoptosome by binding APAF, remember? Activates caspases, activates executioner caspases, and then you have apoptosis. It also releases anti-IAPs that will block the activity of IAPs. What would happen if you don't block the activity of your IAPs? And this all happens. Let's say there's a defect in your anti-IAPs and you activate intrinsic apoptosis. Will that be successful? No, because you'll have these IAPs floating around that are going to bind to your activated caspases and prevent them from functioning. Okay. Survival factors. Okay. So we're talking about activating apoptosis in response to DNA damage or something going wrong in the cell. If it's extrinsic, it'll be in response to a T cell fast ligand interaction binding to an infected cell or cancer cell, signaling the extrinsic pathway of apoptosis. If it's the intrinsic pathway of apoptosis, it'll be due to DNA damage or some drug or something going wrong. And one of the other things that will induce apoptosis is a lack of survival factors. So survival factors are extracellular signaling molecules that inhibit apoptosis, and this helps to regulate cell number during development. So an example is in nerve cells. So you'll have nerve cells and there's target cells that will secrete survival factors, and those nerve cells are going to compete for binding to the survival factors, and only the ones that get the signal survive. The ones that don't get the survival signal, those apoptose. And that's how you regulate cell number. And the way this works is the survival factors are just signaling molecules, and they activate pathways that are involved in regulation of BCL2 proteins. So they're regulating the intrinsic pathway of apoptosis. So one way they could do it is they could, here's your survival factor binding to a receptor, and that will activate gene expression of BCL2, which is anti-apoptotic. So that blocks apoptosis, right? Survival factor, block apoptosis. Also, you could get inactivation of pro-apoptotic BH3 only proteins, right? So you could have BAD, and BAD normally binds to BCL2 and inhibits it, and that's how you promote apoptosis. But the signaling will result in phosphorylation of BAD, which releases BCL2 and keeps apoptosis inhibited. And another mechanism is by inactivating anti-IAPs. Remember, IAPs inhibit apoptosis. So anti-IAPs, if you inhibit them, they would inhibit apoptosis. And that can happen by signaling resulting in phosphorylation of IAPs, anti-IAPs. So they can't inhibit. So no survival factors equals apoptosis. Makes sense because you're surviving, right? Okay, true or false? Survival factors increase expression of anti-apoptotic proteins by binding to a nuclear receptor and activating gene expression. False, right? There's no nuclear receptor anywhere in this. I just wanted to make sure everyone was paying attention. That was a giveaway. An absence of survival factors results in induction of apoptosis via extrinsic pathway. Intrinsic, because it's about regulating BCL2 family of proteins. And so B will be the answer. Inhibition of apoptosis? No, because an absence of survival factors promotes apoptosis. Inhibition of apoptosis in mice only. Well, that's just wrong. And I think, okay, so let me pull up this movie and then we're going to do a practice problem. It's a good movie, so we're going to have to find it. Oh, so annoying. Try once more. Good. It's mostly just like windy sounds. or is it trauma? Okay, so there's pretty much everything. So it was showing the extrinsic pathway, activating the intrinsic pathway, and that does happen. But also the intrinsic pathway could be activated completely on its own. Okay. Okay, so we will finish very early with a practice problem that you guys can work through together, and then I'll help you guys. And then on Wednesday, it's completely review. Okay, so study, bring in your questions, and I will answer them. I will not have any sort of anything planned. I'm just going to stand here to answer questions. Okay. You're studying induction of apoptosis via the intrinsic pathway. You treat cells with UV light because UV is a way of irradiating cells causing DNA damage. Most cells undergo apoptosis within 24 hours. Cytochrome C release is detected as early as 6 hours after UV exposure and can be detected for 10 hours longer. And so the question is whether or not individual cells are releasing a cytochrome C in small bits over long periods of time. So over that 24-hour period, is the same cell releasing cytochrome C? or are different cells being triggered to release cytochrome C? So to look at this, you tag your cytochrome C with GFP so you can look at it, and you observe different cells over 24 hours, and this is what you see. So this is, let me turn my light on. So this is the time. So you're doing live cell imaging. So this is 10 hours, 9 minutes, 10 hours, 15 minutes. So that's one cell that you're watching for that time. This is 17 hours, 10 minutes, 17 hours, 18 minutes, one cell that you're watching. So which model does this data support? A single cell is releasing cytochrome C over 10 or more hours, or different cells are being triggered at different times. So you guys can talk amongst yourselves about this, and then we'll go over it.
Yes. Yeah, I think there's maybe three. And there's one cell.
You know what I mean? So
A lot of quiet. No one has the excuse for quiet but me.
it's been going on. The other cell's doing the same thing, so that's like 8 cents. It's almost done.
Okay, so there's a lot of quiet. Do people understand what they're looking at?
Now I do.
You have no idea what you're
I'm
looking at.
saying now I do.
Now you do. Does anyone have any questions about what the question is or what they're looking at that's preventing them from critically thinking about this? No? Okay, does anyone want to tell me what they think?
You were saying it's probably different cells being triggered at different times because the process seems like it's really fast.
So what in this data supports that?
Well, both the cells, it's only 4 minutes between the first image on the top cell and only 8 minutes between the one on the bottom. So the cell's degrading rapidly.
Okay, so you're saying it's a short period of time between the first and second picture for each cell. So it's probably happening very fast. Cytochrome C is being released very quickly. Now if you compare A and B, what information does that give you to support your hypothesis that different cells are being triggered at different times?
Several hours later.
It's several hours later. So what do you see in the first picture for each one? What are you looking at?
Cytochrome C is lit up.
It's what?
It's lit up, but you can see it.
And so, but what is it? What state is the cell in? What does it represent if you look at this versus this? Is apoptosis happening in the first one or the second one? The second one. Right, so you see how it's these punctate kind of spots in the first? That's cytochrome C inside, where it's confined to the mitochondria. So it actually hasn't been released in the first two. Where it's diffuse and everywhere, in the second two, it's been released from the mitochondria, so it's everywhere in the cell. So now looking at that through a different lens, is what can you say about hypothesis 1 and 2? I think you're still right, but I think the reasoning needs more. So yep.
Well, since the cytochrome C is being released or is still within the mitochondria in figure B at 17 hours or minutes or whatever.
Hours.
Hours? Okay. Since it's still confined within the mitochondria, then that proves that that cell hasn't undergone apoptosis yet. So the cell hasn't been triggered.
Exactly. So notice in both of them in the first panel, you've got cytochrome C inside your mitochondria. It's not released yet. So that completely blows hypothesis 1 out of the water. Because if it was single cells releasing cytochrome C over 10 hours, well, both of them shouldn't have cytochrome C in their mitochondria in the first picture. They should all look like these two, right? Because you'll see it as early as 6 hours. So if it's 6 hours, boom, you see release. They should all have this diffuse staining. But here at 10 hours, and then again at 17 hours, you still see cytochrome C inside mitochondria, suggesting that over time, different cells are triggered. Does everyone follow that? Anyone have questions?
Does it have anything to do with what I was reading about animal cells, most of them needing constant signaling from each other? Does a lack of signaling from adjacent cells or
So
cells?
that's the whole. You're going into survival factors. So animal cells needing constant signaling from each other to stay alive, to survive. This is different because here what you're doing is you're treating cells with UV. So it's just that over. Okay, so are you saying that maybe they're withdrawing survival factors? Because I'm trying to connect UV and survival factors. And why? So why would this be happening is the question, right? Why would you see? Because if you have a layer of cells, you treat them all with UV at the same time. Why don't they all just, boom, release cytochrome C? Why is it happening over long periods of time? And so what I'm hearing here is that there's some sort of signaling from other cells that maybe is activating apoptosis or possibly inhibiting survival or something like that. I think that's a very logical hypothesis. Because it doesn't actually, this question really doesn't go into why, but you should always be thinking why. And so that's the question is like, if it's just DNA is damaged, why? Well, so let's go a little further, okay? So if DNA damage is causing apoptosis, how does DNA damage cause apoptosis? Can anyone just speculate? Yep.
Proteins like say maybe your IGs like might not be made in.
Go back a bit. Go broader. You're stuck in apoptosis. Go really broad. What happens over a 24-hour period that might sense DNA damage? Yep.
If like it starts to, the DNA starts to acquire like mutation, and therefore it can't like replicate its DNA, can't function properly. And that's what would cause it to like.
Right. So you have DNA replication that happens, right? If you've got mutations in your DNA, will DNA replication happen? Okay, so let's go back to checkpoints and cell cycle. Now we're going to integrate this all together. It's going to be painful. We're going to do it though. So you're right, but let's go a little bit further. So you've got S phase needs to begin, right? But what's going to prevent S phase from beginning? Right? If you've got DNA damage, you shouldn't be replicating your DNA because there are checkpoints in place to stop that. And let's say you do replicate your DNA because you get through it somehow, because that happens too. There are more checkpoints that should prevent you from going through mitosis completely. So those are two things. We learned about this. We learned about checkpoint activation a little bit. We learned about MYC and we learned about P53 and all these things that signal apoptosis, right? So that's really is what's happening here. So you're irradiating your cells, damaging DNA, activating checkpoints, activating these signaling pathways that turn on P53, right? And P53, what does P53 do? One of two things. Induces apoptosis or cell cycle arrest. So why would you see this happening at different times? Why are different cells going through apoptosis at different times in response to UV damage? Yep.
Could they be at different places in the cell cycle?
Yay. I love it when people get it right off the bat. Yeah, well, I mean, I didn't do the research myself, but that would be my thinking, right? There are different places in the cell cycle. Do cells all divide? If you have cell culture, they're not synchronized, right? They're all going through different points in the cell cycle. So chances are it takes time for them to get to a point where they're like, oh no, damaged DNA, activate P53, activate apoptosis, right? Good. Is everyone following? Yep.
Could you also argue that maybe it's just unequal distribution of DNA damage, like maybe it takes longer for certain cells to get damaged?
Unequal distribution of DNA damage. It's possible that some cells just have really horrible DNA damage and they're going to undergo apoptosis quickly. But I like the cell cycle thing better because you have to think about detection. When is damaged DNA detected? I didn't teach you that in this class. Those who have taken 409 should know this, but things like things that prevent replication. So let's say you're replicating your DNA and you've got a big bulky lesion or a thymidine dimer or something that's going to stall your replication fork. Things that like slow down the process of replication, those are the things that trigger the DNA damage response in apoptosis. So I think it's more likely that has to do with sensing, and most things are sensed around the time of S phase. Any other questions? Is everyone following this? Okay. So if you wanted to study the mechanism, like Danielle brought up, she was wanting to know why is this happening like this, and we just talked through it and came up with some ideas. What kind of experiment would you do to see whether or not these cells are at different phases of their cell cycle? Yep.
Couldn't you use temperature-sensitive mutants to stop the cell cycle at the same time and then induce damage?
So figure out a way to synchronize your cells, perhaps, right? So if you use a temperature-sensitive mutant that has a mutation at some point of the cell cycle, it will halt the cell cycle. You can do that, turn up the temperature, arrest your cells, turn back down the temperature, let them go again, induce damage. And what should you see if you induce damage? So now you have synchronized cells in your culture and you induce the damage. Will it look like this, or do you think things will look differently if it's just based on where you are in your cell cycle?
Different.
It'll look different. How do you think it'll look?
I think that if they're all synchronized cells, we said, right? So then it would all be happening at the same time instead of over different amounts of time.
Right, so exactly. So if we have synchronized cells and we induce DNA damage, they should all be hitting different points in the cell cycle at the same time, right? And we should be seeing induction of apoptosis at the same time. And so I'm guessing if you took pictures of the same cells, you could take pictures of your whole culture, because you can see even just in this picture, this one's undergoing apoptosis and this one's not, right? Same here too. This one is, this one's not. And you should start to see the whole plate become diffused and stained like this at the same time. So good. And people do this a lot when they're studying cell cycle and apoptosis and anything involving cell cycle. You can use drugs. You don't need a temperature-sensitive mutant. You can use a drug that will halt your cell cycle at a certain point. So it will create, it'll activate a checkpoint. Then you remove your drug, clean your cells, let them recover, and then they'll all start replicating in the same cycle. Any questions about this stuff? No. Okay. Well, that's it for today because my voice can't take it anymore.